Okay, welcome everyone. Good afternoon. So today it is my greatest pleasure to introduce this week's seminar speaker and this year's Wagner lecturer, Dr. Emily Sessa. Emily, she got her PhD from the University of Wisconsin-Madison, and she has served as an associate professor at the University of Florida until 2022. When she took a position as the director of the New York Botanical Gardens, which is, for those who don't know, the largest herbarium collection in the Western Hemisphere, an amazing place to work at. And I think this is especially cool because that she is here today, because Wagner, the person who endowed this funds for this seminar, was a fern specialist, and Dr. Sessa, she's also a specialist in ferns and lycophytes. So I think Wagner would be super happy with our choice of inviting her today to give this talk. So without further ado, let's welcome Dr. Sessa.
Thank you. Thank you, Thais. And yeah, thanks everybody for having me. I'm really excited to be here. Hopefully you can hear me. Good. Okay. I'm getting over a sore throat situation, so hopefully I've got water here, I have tea, I have cough drops, I have all the things. So thanks so much for the invitation. It's really a tremendous honor for me to be here. So as Thais mentioned, and as you can see, I'm at the New York Botanical Garden now after about a decade at the University of Florida. So I love just showing this picture because it's a beautiful picture of New York Botanical Garden and our conservatory, and you can see the Manhattan skyline there in the distance. So my position at the garden is director of the herbarium. So I think about the herbarium most of the time these days, but when I'm not thinking about the herbarium, I'm thinking about ferns. So ferns are the group of organisms that I am the most passionate about. They captured my attention and my love quite early on in my career, and I've never looked back. They're really wonderful, beautiful, and very interesting in a lot of different ways. And of course, they were also the favorite of Herb Wagner, who deservedly very, very famous in the fern community. So it's really a tremendous honor for me to be here today giving the Wagner lecture. Herb worked on many things, but he's most well known in the fern community for his work on Batricium. So here's a little Batricium to show you, a little fun little plant. And this is a great picture of Herb and Florence in the field photographing a Batricium. And of course, as you know, Florence was his partner in crime and a formidable botanist as well. So really, really special for me to be here today. And ferns are wonderful plants. They're often very overlooked. So if you've heard of, plant awareness disparity, I think ferns suffer from that even more than most plants, because even among botanists, sometimes they don't get the attention that I feel they deserve. So if you go out in a lot of different habitats, though, ferns are there, and they're playing very important roles in ecosystems. This is an example of, I'm going to point only at the center screen so that I don't make myself seasick here. But this is a hammock in Florida where you can see that basically everything in the understory here is a fern. So there can be incredible fern diversity. This is in not even far enough south in Florida to be truly tropical. So even in sort of subtropical habitats, ferns can be incredibly diverse. They're really incredible dispersers, so they often are, I was talking about this earlier with someone, often ferns are kind of pioneer species. They're the first species that might colonize a new habitat, like new lava flow in Hawaii, which is what this is showing you here, and you can see the ferns very happily colonizing this new exposed substrate. This is a fern in the desert, so contrary to sort of common thought about ferns as being only living in dark, shady forest understories, they can actually be quite diverse in terms of the habitats that they can occupy. And there's a whole family of ferns, the Pteridaceae, that has evolved specifically in desert habitats, or many of its members have evolved in desert habitats. The other end of the spectrum, they can also, some of them do quite well in water. So this really bright green thing here is Ceratopterus, and it's growing also in Florida in this sort of floating kind of mat situation. And there's also a group of ferns, a very small order of ferns that is entirely aquatic, and they're called the water ferns. So there really is a huge amount of diversity in these organisms. It makes them a lot of fun to work with and to study. So in terms of sort of numbers, just a very brief overview here. So the names of these things. Ferns have been considered a class. They've been considered, sort of different levels here. But this is the current classification system, which is the pteridophyte phylogeny group. And this is the equivalent of the angiosperm phylogeny group, which was established a long time ago. And it took until 2016 for ferns and lycophytes to get kind of the equivalent of that classification system. So in our current understanding, there are 4 subclasses, 11 orders, 48 families, about 300 genera, and about 10,500 species. So a reasonable amount of diversity. And after the flowering plants, ferns are actually the most diverse of the vascular land plants. There aren't that many options to choose from, but you go from flowering plants to ferns and then to the gymnosperms and the lycophytes. So second most diverse among the vascular plants. So brief overview of what I'm going to tell you about today. So we're going to have kind of 3 parts to the talk. I'm going to start off with a bit of background on fern phylogenetics to provide some context for the questions that I'm going to be talking about towards the end of the talk. And then I'm going to tell you a bit about the GoFlag project, which is a big project that I worked on for a number of years and was a PI on during my time at the University of Florida that built the probe set that was used to generate all the results that I'm going to be talking about today. And then I'll tell you about the results of that project for ferns specifically. So starting with the kind of background section, I wanted to give you kind of an idea of what some of the questions in fern phylogenetics are that have occupied the fern community for a really long time. And if these names and words mean nothing to you, don't worry, I will show a lot of pictures. So the fern phylogeny that I'm showing you here is from the pteridophyte phylogeny group. The names are probably quite small and you might not be able to make them out. But there are 3 sort of major questions that fern folks have been interested in for a long time. So one is the monophyly of a big group called the leptosporangiates. Then the next is the relationships among several lineages that are called the eusporangiates. And then if you've ever seen a horsetail, the relationship of horsetails in ferns has actually been quite contentious for a long time. So this big group here in blue, that's the leptosporangiates. Then we've got 3 groups, and it actually includes the horsetails as well, that are these eusporangiates. And then the horsetails are shown up there in green. And I'm going to just blow up this top part here so you can see a little bit better what's going on. So basically we have what we think is this enormous clade that we call the leptosporangiates. And then we've got these 3 different groups here that I've got in tan, and then we've got that group of the horsetails. So how these groups were all related to each other has been open questions that the fern community has been interested in for a really long time. So the leptosporangiates, this includes a number of orders, bunch of families, and almost the entirety of the species level diversity. So this is a really large group, the leptosporangiates, more than 10,000 species. The eusporangiates are 3 separate lineages that are now recognized at the level of order, and they're modest in size compared to the leptosporangiates, but they have a lot of morphological and habit and habitat diversity. So this is one of the Moradiaceae members, and you can see that it's kind of tree-like. It's not a tree fern in quotes. Technically, the tree ferns are this group here that's within the leptosporangiates. But size-wise, they're giving the tree ferns a run for their money. They're sort of on the same scale. Then in the middle here, this is a member of the Ophioglossales, and maybe you can make out these sort of pointy structures right in the middle here. So these ferns actually have a leaf that has 2 separate sterile and fertile components to it, which we find that sort of sprinkled throughout the fern phylogeny, but it's kind of a unique feature of that group. And that includes the batriciums that Herb loved so much. And then over here, this weirdo is the whisk fern. This is in the group Xylitales, which is quite small. And this plant is unique and interesting because it doesn't have true leaves, it doesn't have true roots. The homology of some of its structures with other ferns and other plants is very difficult to figure out and actually still sort of under question. So these are 3 very sort of diverse and bizarre groups. And so how they're related to each other, how they might be related to the leptosporangiates or to the horsetails, interesting open questions for us in the fern community. And then the last of these here is the horsetails. So this is another situation where, based on morphology, you would not be inclined probably to say that these were ferns, right? They don't look very fern-like. Their reproductive structures, their spore-bearing structures, are in these strobili, little cone-shaped guys. These are actually the leaves that you see here. The leaves are highly reduced, and they form these little sheaths at the nodes. And so this is a highly branching species that I'm showing you here, and these are actually all branches. They're not leaves. So these things that come off to the side, the leaves are these things. And the horsetails have a really interesting fossil record, and so placing them in relation to extant plants has been very challenging, and as I mentioned, still sort of contentious. And so extant fern people tend to call horsetails ferns, but paleo people tend not to like us doing that. So how they were related to the other ferns, they are eusporangiate, so it seems reasonable to assume that they would be somewhere near them in the tree, but these have all been open questions for a long time. So the first non-morphology-based kind of attempts to answer these questions came in the early 1990s to early 2000s. So this was when chloroplast DNA restriction site mapping came online, and the first sorts of sequence data that were also from chloroplasts came around. So see, these are some of the kind of classic papers in Fern phylogenetics from that time period. And they were able to pretty confidently say that yes, all those leptosporangiates, they are monophyletic. That does make a good, big, well-supported clade. But the answer to these other questions was sort of equivocal. It wasn't really clear based on those early data what the answers to these other questions were. And I really love this. This is a little figure from one of Kathleen Pryor's papers in the early 2000s, where you can see there's a lovely polytomy there of how these things were related to each other. And then the ophioglossoids and the whisk ferns, that's where Batricium lives, and that's the whisk fern. Those two things are always very close together, but even this relationship was a bit tenuous. And so all of this basically is sort of up for grabs in terms of figuring out the answers to these questions. So from that time to now, these have been sort of the major themes and questions in fern phylogenetics. And especially as we've gotten more towards the present, this first question has become more and more interesting. So what can we learn from plastid genomes versus what can we learn from nuclear genomes? I'm not going to talk too much about this aspect of nuclear genomes today. I'm going to keep it relatively simple, but fern genomes are incredibly complex and complicated. So ferns are well known to like to hybridize, to like to undergo polyploidy. Average chromosome numbers in ferns are in the 40s. Average genome sizes are 12 to 13 or 14 gigabases, haploid. So you can do the math for whatever group you work on. I bet our genomes are bigger than yours. So they have really big, complicated genomes. That's super interesting, but I'm not going to talk too much about that today. So what can we learn from plastid versus nuclear genomes is increasingly of interest. Then we've got these sort of continuously recalcitrant nodes. So that includes some of the ones I just talked about, and then some others in leptosporangiates that I'll get to later with some interesting relationships that we've wondered about for a long time. And then, of course, I have these little illustrations I always forget about. And then sort of dates, rates, and other kinds of add-ons. So what can we do with molecular dating, and how can we tie that to things like biogeography? Rates of evolution tend to differ across the fern phylogeny as a whole. So how can we bring some of these new data types to bear to answer these sorts of questions, or to ask questions that are in these sorts of areas informed by those types of data? So now I'm showing you a rather overwhelming table. I don't like a lot of text on slides, but I'm doing this for a reason, and so I'm going to walk you through this table because you're going to see sort of a condensed and colorful version of it later on in the talk. So what I'm showing you here is a list of all of the what I'm calling comprehensive studies on fern phylogenetics. And what I mean by that is these are all of the studies that had enough taxon sampling to be able to get at some of these really deep questions in fern phylogenetics. So from horsetails through leptosporangiates, asking sort of deep questions in that phylogeny about relationships. All of the papers in green used plastid data. There's one mitochondrial study to date on ferns, and then all the ones in these sort of blue and purples at the bottom are studies that use nuclear data. Over here, you can see how many, and they're organized by date within each of those categories. So 2004 to GoFlag, which is what I'm talking about today, 2015 for the nuclear data down to GoFlag. And you can see over here, more or less, there's been sort of a steady increase in numbers of loci that these studies have used, the plastid ones in particular. And then there's also been kind of a steady increase, although with a few, kind of dips in the numbers of fern taxa that they included. And so those are just the numbers of ferns. Outgroups are in addition to that. So some of these have been really large in terms of the taxon sampling. So we've got a 2011 paper here that had 2,600 species, a 2018 or 2016 paper that had nearly 4,000 species, but were based on only a couple of plastid loci. And then you get down here to the more recent ones, and we've got almost all of the plastid loci with some larger numbers of species. And then on the nuclear side, also sort of increasing numbers of species and a movement to use whole transcriptomes, or in the case of GoFlag and this other paper, low copy nuclear genes. So what I'm going to show you later in the talk is going to be a version of this table that's condensed to look like this up here. So the individual cells are going to be corresponding to the different papers, and then they're going to be colored in accordingly. So I'm going to use this later in the talk, and I want to show you an example of it here so that we're all on the same page, and we'll come back to it. We'll talk about it again later. So when you're testing different topologies and you want to know whether a data set confirms or conforms to a particular topology, that's how I'm going to use these tables later. So if you think about these three tips, for example, Equisdales, other ferns, and seed plants, there's three possible relationships of those tips to each other. And if this table was filled in like that, it would mean that all of these studies up here found this resolution of that relationship. So Equisdales sister to all of the other ferns, and then the seed plant sister to that little group, as opposed to these other two resolutions of those tips. Does that make sense? So you might be tempted to try to like trace the tips over and make them correspond to lines or to rows in the table. Don't do that. That's not what we're doing. I know that's a confusion. All that this is saying is that all of these studies, because all of these boxes are colored in, had this resolution. The alternate might look like this. So if you saw this, what that would mean is all of the plastid studies, which are these green ones up here labeled plastid, all of the plastid studies had this resolution, and then the 4 nuclear studies were split between these 2 resolutions. Good? Okay, we're going to see a lot more of these later, and the tables are going to be a lot bigger. So I like to show a little toy example first, just to get you acclimated, as it were. So that's the end of my kind of fern introduction part. And next I want to talk about GoFlag a little bit. So the GoFlag project was funded under the Genealogy of Life program at NSF. This was a relatively short-lived program. It was supposed to last for 10 years. It lasted for only 2 or 3, but we were lucky enough to get funded in that short amount of time. And NSF had conceived the program's purpose as illuminating the dark parts of the tree of life. So they really wanted to encourage large-scale studies on parts of the tree of life that PIs could make a case had been understudied. So, you know, taking a dark part of the tree and illuminating it. And the GoFlag consortium was a set of faculty from the University of Florida. We were the lead institution, and then some folks from the Field Museum and the University of Arizona. And we had an undergrad REU who designed this really fabulous logo for us. So if you think about the land plant phylogeny, so this may or may not be accurate as the outgroup. You can ignore that down there. But all the other land plants up here, so we've got our angiosperms and gymnosperms, our seed plants, ferns are sister to them, then the lycophytes, and then we've got, showing here as a clade, the bryophytes. And thinking about this phylogeny as a whole, flagellated sperm are the ancestral condition for these plants. So if you look at, these are some algal sperm, this is a liverwort, a fern, another fern, a cycad. You can see really beautiful flagella in various configurations on all of these guys. But then when you get to the angiosperms, they are their sperm, and they don't have tails. So there's no flagella on the angiosperm sperm. And so the flagellate plants are essentially all of these groups here except for the flowering plants. And GoFlag was focused on phylogenetics of all of those groups except for the flowering plants. So it's not a clade, obviously, because we're missing one rather important character up there. But it included everything else that you see here in kind of the slightly bolded font. Now, for the plant people, a note on the gymnosperms. Some of them have flagellated sperm, some of them don't. So cycads and ginkgo do, but the other ones, the conifers and the neophytes, do not. But we wanted to be inclusive, so we included all of the gymnosperms as sort of honorary members of our sampling. It just didn't make sense for the purposes of this to exclude just a couple of those of these groups. So why focus on this group? Why target this set of lineages for any sort of phylogenetic analysis like this? And what compelled NSF to give us this money? Well, the argument that we made was that if you look at the evolution of land plants, you will find numerous major innovations in land plants that would have happened in the ancestral flagellate plants. So for example, the movement onto land, rather important, would have happened in a flagellate plant ancestor. The first fungal symbiosis also would have evolved then, so not in any particular order, of course. Stomata evolved in the ancestral flagellate plant. Vascular tissues, roots, stems, all of these things. The shift to a sporophyte dominant life cycle from the bryophytes to the tracheophytes up here. The first true leaves would have evolved in this group. Seeds as well in the seed plants, because of course we've got gymnosperms here. Secondary growth as well. A couple of evolutions of heterospory in different places. So the seed plants, of course, are heterosporous. It's a prerequisite for seeds. And then both the ferns and the lycophytes have groups within them that are heterosporous. And that's tied to a lot of different things having to do with chromosome numbers and genome sizes and complexity and things like that. And then a number of other features, like the first transitions back to aquatic habitats, would have happened in flagellate plant ancestors at some point in the backbone of this tree. Parasitism presumably would have evolved first then. Epiphytism likewise. The independent diploid sporophyte that kind of goes along with this shift to the sporophyte dominant life cycle. Animal-mediated dispersal, pollen and pollination. So all of these different things, flagellate plants would have happened or would have been the ancestral taxa in which those things evolved. And if you want to study some of these things in angiosperms, you have to use these groups as one or more of your outgroups. So to provide those sort of evolutionary comparisons for thinking about the evolution of any of these traits and this sort of a timescale, you really need data from flagellate plants, and you need accurate information about their evolution and their relationships to each other. So that was the argument that we made in our proposal that eventually was funded for GoFlag. So I'm not going to go too deep into the methods, but I want to just give you an overview of how we approached this. So we developed a probe set for doing targeted enrichment of nuclear loci. So we used what at the time was all of the available transcriptomic and genomic data. So practically, this was mostly data from the 1KP project, which the plant folks in the room have probably heard of. This was a major initiative to sequence 1,000 plant transcriptomes. Now it doesn't seem like that big a deal, but back then it was a big deal. And so that project had almost 1,200 green plants, including 241 flagellate plants. So we included all those transcriptomes. And then a few more land plant genomes, mostly not ferns, because at the time there were no publicly published fern genomes. We identified 410 single or low copy nuclear genes in that set, and then looked for exons in those genes that were at least 120 base pairs long, and they had to have 65% identity across at least 6 of those genomes. So we were really looking for loci that were going to be conserved across this really massive span of evolutionary time. We ended up with 450 quote-unquote loci, which really are exons. So when I say loci, what I mean are exons. And we did a pilot study and then optimized the probe set even more. And this was the taxon sampling for that pilot study. So it was heavily biased towards liverworts and ferns, because that's what people in the PI group worked on. But we did, and mosses as well. But we also had some hornworts and gymnosperms and lycophytes in there. So this optimization allowed us to get rid of loci, most of the loci that really only worked well in one or a few of these groups, so that we were left with loci that worked well across the board. So the end result of this was called the GoFlag 408 probe set. So it was 408 exons. And there were about 52,000 probes covering those exons, and those exons were located in 229 genes. So multiple genes would have had multiple exons within them. So if you want to know more, this is the paper that published the probe set and all of the methods. It was published in 2021. So welcome to go look up the paper if you're interested. And the one figure that I wanted to show from the paper was, from that paper, was the distribution across loci and across taxa of successfully sequenced loci. So what this is showing you here is that there was a relatively small number of loci that were recovered only from a small number of taxa, if that makes sense. So the number of loci recovered ranged from 3 to 436 out of 451, which is pretty good. Average of 332, median of 394. And I think I have here, so fewer than 10% of the loci were recovered in 16 or fewer samples. So it's good, basically, that these bins over here are relatively short. And it's good also that this bin over here, which is a lot of loci, is high, meaning that a lot of loci were recovered in a lot of taxa and relatively well distributed across the lineages. And of course, like blue is gymnosperms, so they were a bit more variable. But the hornworts, which is up here in brown, they did pretty well, like most things sequenced in hornworts. So hopefully that makes sense from this figure. And the impact of GoFlag is something that I'm really, really proud of. So when we conceived of this project, the model that we decided to use was basically to community source the sampling. So we were going to develop the probe set, but then we reached out to, as much as we could, the entire sort of global community of people working on these groups, especially students, and we said, hey, we have money, we have like $2 million to sequence stuff, send us your stuff, and we'll sequence it for you. And we will give you the data back for your purposes to do whatever you want with an understanding that, you know, after a few years, we want to make all the data public so that it's publicly available. So we basically funded the PhDs of a number of graduate students around the world. And so far, I think there have been 70 or 80 papers published and many, many, many abstracts and talks at conferences, and still counting. And so these studies range from family level studies. So this one on Phylogenics, this was a PhD project. Glyciniaceae, Schiesiaceae, so some family level analyses. Then there are some that are more genus level, so in mosses and some other groups. In a fern, this was actually a fun one where I used the probe set to identify a new hybrid fern. And then there have been some much bigger ones as well. So this was published in 2023, and it's a time tree of all bryophytes. And I actually got an alert from the American Journal of Botany the other day that apparently this was the most so far is the most highly cited paper from 2023 in the journal. So that's cool. And there's another one that's hornwort specific right now that's in revision. So this is probably in my whole career, like the single project I'm most proud of because of the impact that it's had, especially for students in various places. So now moving on to the Fern results from GoFlag. This is the highlight. And I said I don't like text, and then I've been showing you a lot of text. I promise we're gonna move back to pictures in a few minutes. The ferns, just so you know what we're talking about in terms of the fern data set, we were able to include almost 1,000 ferns, plus additional outgroups from the lycophytes and the seed plants. The matrix of all of the nuclear data was about 77,000 sites, third of that for amino acids, and we had pretty good taxon occupancy, so about 90%. 202, 203 taxa out of the 220, genes out of the 229. And I want to just say that this is another way of saying that we did not have very much missing data. So we applied really stringent filtering criteria to filter. We didn't want a lot of missing data. And these were, this taxon occupancy for genes is that these were the ones that passed our filtering criteria for being present across taxa in the matrix. So we analyzed these data in a number of different ways, and we were interested in the effects of different substitution models on all of the gene trees. So we did nucleotide, codon, and amino acid models, and that's all going to be in the paper. That's hopefully going to be submitted in the next couple of months. But today, what I'm going to show you is the species tree that resulted from the best model for each gene tree. So we ran all these gene trees with all these different models, but for each gene tree, we just took the best performing one and put those into Astral to produce a species tree. And that species tree is what I'm going to show you here. We also assembled plastid data from the off-target reads, and that's how we got the 79 assembled plastid loci. And it was sort of funny, at the beginning of this project, the company that we were working with, these off-target reads, as far as they're concerned, they're junk. It's extraneous to what they want, which is the targeted nuclear data. And as the project went along, they would come to us really excited because they had improved the efficiency. And we were like, no, go back to being inefficient because we want those plastid data. We want those off-target reads, actually. They're very useful for us. So that was a bit of conflict with the sequencing company that we worked with. So this is that species tree. So this is the GoFlag Astral species tree. And all of the branches received maximal support except the ones that I've noted with the numbers here. And these are local posterior probabilities for the primary topology. So you can see there's pretty good support. Even the ones that are numbered, those are decently high. We've got a couple in here that are low, and not surprisingly, those are short branches. But even some of the short branches still have pretty high support. These pie charts are showing you the quartet support. So again, at every node, we've got 3 possible topologies. And where you see green, that is agreement with the species tree topology. And then you can see the 2 alternate topologies there. And so these pies on this side are showing you the support for all of the branches along the backbone. And then these pies are showing you the support for the crown node of each family or order, depending on what is shown. So in some cases, it's orders like Cyatheales and Salviniales. In some cases, it's families like Saccharomyces. And then the U polypods up here, that's a really gigantic group. So if you think back to the leptosporangiates that I was talking about before, the leptosporangiates start right here. If I kind of draw a line under Osmondaceae, everything from Osmondaceae up, this is all the leptosporangiates. So again, 10,000 something species. But then within the leptosporangiates, the U polypods is a group of like 20 families. So it's quite large. But the questions that we were really interested in all concern these sort of groups that are kind of successively sisters somewhere along the line to the U polypods, or these groups down here that I've already mentioned. So these U sporangiate lineages down here towards the bottom. So you can see like most of the families have there's pretty strong concurrence with the species tree topology, except in a couple of cases. And again, along the backbone, decent support, decent agreement with the species tree, except where you have really short branches. And that's where you start to see a split of support among the different alternative topologies. So next, this is the nuclear and plastid trees facing each other, looking at each other. So this is that same nuclear tree that I just showed you. And then this is the plastid tree. So the nuclear side, again, this is the species tree from Astral. And then on the plastid side, this is based on, this is an IQ tree analysis of all of the concatenated plastid loci. And what I've done here, so same thing, that all support is maximal except where you see the asterisks, and that's indicating less than 100% support. So it's whatever the value is that's appropriate to the analysis. And I've highlighted in black on the topologies, and then in the middle here with these sort of crossing lines, places where there is conflict between the nuclear and plastid trees. So you can see there's kind of 3 places that stand out. So the Cyatheales and the Salviniales, their relationship is different on the nuclear side than on the plastid side. And then same down here with the Hymenophyllaceae and Glycinaceae, and then with our eusporangiate groups, which is really annoying because, spoiler alert, the answer is that we still don't really have a firm across the board answer to what's going on with the relationships of these 3 groups. But I'll talk more about that in a few minutes. So what I want to do next is walk through these parts of the fern tree that have historically been difficult. So I've already mentioned this one down here, the eusporangiates, and I didn't mention these other ones earlier on, but these are additional kind of recalcitrant nodes that have been very difficult to resolve using just plastid data or morphology sort of based analyses. And this one up here, the Denstedtiaceae and the Pteridaceae and how they relate to the eupolypods, these actually have the same relationship on our 2 topologies, but it has been an area of contention in fern phylogeny. And so I wanted to highlight that as well and talk about that one as well. So I'm going to go back to the complicated table that I showed you earlier. And obviously you remember all the details of everything that was in that table. Of course you don't. So I'm just going to show it again real quick. So again, plastid loci kind of generally increasing, and taxa sort of increasing, but not in any sort of linear way. And then most of those nuclear studies use transcriptomes, except for, we had these low copy data. And then GoFlag had the highest sampling in any of the nuclear studies. And I'm going to go back for 1 second just to say that these run oldest to more recent, and then it kind of jumps back up here. And so this is the most recent. This is GoFlag. There's that mitochondrial analysis, and then it's the same with the nuclear data where the oldest paper is here, goes down, and then kind of hops up and then goes down again. So we'll take these one at a time, and I want to start with this one, which is this conflict between the Cyatheales and the Salviniales. And in some ways, this is, if you know ferns, this is kind of interesting that there would be any sort of difference in relationships between how these groups are related to this group up here. And the reason I say that is these two orders almost couldn't be more different. So the Cyatheales is the tree ferns, the true tree ferns, and the Salviniales are the water ferns. So these include floating aquatics that are very, very small, which is a zola that I'm showing you here. This is a Salvinia species. There's some interesting morphological variation in this group. It also includes Marsilia, which look like four-leaf clovers and are rooted, but they're always associated with aquatic habitats. So morphologically and habitat-wise, these two orders they're just as different as night and day within the ferns. The other thing that's really interesting is that the Salviniales is the one group within the ferns that is heterosporous. So all of the other ferns are homosporous, and that has implications for genome size, reproductive system, and mating system. So whether the Salviniales are sister to this whole group, which is suggested on the nuclear phylogeny, or whether the Salviniales and Cyatheales are sister to each other, which is what the plastid data found, that just sort of has interesting implications for that evolution of heterospory, and how we might want to think about this group up here that is the sister to either that little clade together or to just the tree ferns. So very intriguing. And this, I'm starting with this one also because it's sort of the easiest to understand my little tables, if you will. So I'm not showing all the possible topologies. I'm just showing you the two topologies that were found in these data sets. And almost all of them found this relationship, which has the tree ferns sister to the Polypodiales, which is this whole group up here. So that's this, with the water ferns then sister to that clade. And there were two studies that had the tree ferns and the water ferns sister to each other. One of them obviously being GoFlag. So these two were the two most recently published chloroplast analyses that both found that pattern, different from all of the previous plastid analyses and all of the nuclear analyses which found this relationship. And interestingly, these two papers are the only two that have essentially whole plastid genomes, and they had the largest taxon sampling of any of the plastid analyses. So if you think back to the table, all of these others, some of them come close, but have very few loci. So these were the only two that had the combination of highest taxon sampling and highest locus sampling, marker sampling. So I don't know what to make of that. It's very interesting. So clearly there's some, like, it depends on who you sequence and what you sequence potentially. And I don't know that I'm confident saying this is what's true, number one, just because the balance of evidence agrees with it. I think this here, what I have in pink, is a compelling reason to consider the possibility of this topology. And also there's for a lot of these relationships that I'm going to show you, these ones that are in gray, the branches are really short, right? So there's the possibility for there to be incomplete lineage sorting. There's the possibility for there to be a hybridization event involved that could have involved chloroplast capture. So that might be where this signal is coming from. So hypotheses to be tested with additional data, although I don't know what additional data you would gather. Maybe it's going to be structural or chromosome level assemblies or something like that, even more plastid sampling. So it'll be interesting to see where we go to try to resolve this that seems unresolvable at this point. So the next one I wanted to mention is up here at the top of the tree. So this is, the question here is, who is the sister to the eupolypods, this very diverse group? Is it the Denssteadiaceae or is it the Pteridaceae? So these are not as remarkably different as the last two groups that I showed you, but they do have some unique features. So the Denssteadiaceae, this includes bracken fern. If you're familiar with any fern, it's probably bracken fern. And this family has, some of their species have these sort of unique cup-like sori, which are their reproductive structures, their spore-bearing structures. And then the Pteridaceae have these really beautiful linear sori that tend to be found along the margins of the leaflets. And which of these two groups is the sister to the eupolypods, it's just been an open question in the fern community for a really long time. So we were eager to see what the data said about that relationship. So here, three possible topologies. So either the Denssteadiaceae is sister to the eupolypods, or the Pteridaceae is sister to the eupolypods, that's number two, or those two form a clade, and that clade is sister to the eupolypods. And we have all three of those possibilities present in our set of studies, our set of data sets. So here, the pattern of interest is that all of the nuclear analyses support dense steady AC being sister, and the plastid analyses are all over the map. So it depends on what you sequence, depends apparently on who you sequence, as to which of these resolutions you get. And the one mitochondrial analysis agrees with a few of the plastid analyses. And in this case, the GoFlag analyses both agree with each other, so I'm calling it case closed. I think dense steady AC is sister to the eupolypods, and I never want to hear about this question ever again. So that's like one that I feel, obviously I'm being a bit facetious here, but I do feel, sort of reasonably confident that and that NIDA et al. Analysis that's right above GoFlag also agreed with GoFlag in this case. So I'm decently confident that we can say that the dense steady AC are the sister group to the eupolypods. Next, let's jump down here to the bottom of the tree. I know you are all just waiting with bated breath for me to get to this part since I made such a big deal of it at the very beginning. So here we've got our horsetails and then our other 3 eusporangiate lineages. And again, just to remind you, we're talking about morphologically very different organisms here. So how are these things related to each other? The Ophioglossacea and Silitaceae, which are often also recognized at the level of order, these two, they always go together. So there's clear support for a sister relationship between those two. So I'm not going to show you any alternatives that don't reflect that relationship because they don't exist in any of the data sets. So I framed this as sort of being around equisetum because, like I kind of alluded to earlier, whether or not equisetum is a fern has been a long question. And if you go back to some of those sort of early papers in the modern phylogenetic age that looked at these relationships in ferns, it was a big deal when they figured out that horsetails are the sister to ferns. And that's what we've assumed to be the case based on those early studies, that equisetum is the sister to all of the other ferns. So we were really interested to see whether we found support for that relationship. So I'm showing you first, these are the 2 scenarios in which the horsetails are sister to all of the other ferns. So the relationship within the other ferns then varies a little bit with whether the eusporangiate lineages form a clade, which they do in scenario 2, or whether they do not form a clade, basically. And the Miratiales forms a clade with the leptosporangiates. So you can see there's a lot of boxes filled in, and I should mention that where you see the dashed line here, that indicates that that study did not have the appropriate sampling for us to answer this question. So that's all that means, is that that study is uninformative when it comes to this question. So similar to my toy example I showed you in the very beginning, the nuclear studies all agree that equisetum is sister to the ferns, but they're split then, oops, sorry, wrong button. They're split in whether they support a eusporangiate clade or not. But all of the nuclear phylogenies support equisetum being sister. And the GoFlag plastid data also support that. But if we look at all of those other data sets, so there's GoFlag. So plastid and nuclear data are equivalent, or sorry, they're split in whether they support those 2 eusporangiate scenarios, but at least they agree that equisetum is sister. But then if we think about the cells in the tables that are not filled in, that's because there are a number of other scenarios that we find in all of some of those other studies. And so these other 4 that I've just added, the difference is that equisetum is now no longer sister to the rest of the ferns. It's somewhere nested within these other fern groups. So there's not a huge amount of support in terms of numbers of studies that find that relationship. But the one I want to point out is number 3 over here, where that green one that I've got the pointer on, that's the NIDA study, that's the other large plastid analysis. So we've got now a difference in plastid support between a couple of very different scenarios between those 2 studies. So this large study, ours from GoFlag, finds this relationship, and that other large recent plastid analysis found a pretty significantly different set of relationships among these lineages. And then these down here, I'm including them because they are, there is support from them in these studies, but these are the older plastid studies, these 3 that I'm kind of highlighting here, that tended to have lower numbers of loci and lower numbers of taxon sampling. So I'm pretty confident that our answer to this set of relationships lies somewhere in this part of the slide. but not totally clear which I want to place my bets on at this point. So again, still more work to be done here. And then finally, the last one I want to talk about is this one, kind of right in the middle. And you'll see that the conflict here involves Hymenophylaceae and Glyciniaceae, but Dipteridaceae and Metonia are also sort of included in this scenario because how you resolve this relationship has taxonomic implications. And that's the first of these kind of historically challenging places in the tree where there are significant implications for our classification system based on which resolution you adopt as being true or real. So I've got Glyciniales here in quotes because Glyciniales, when it is recognized as an order, includes Dipteridaceae, Metoniaceae, and Glyciniaceae. And if you look at this phylogeny, you'll see Dipteridaceae and Metonia here. They always go together. So this is another situation where those two are always each other's sister. And then here you can see Glyciniaceae is sister to those two. So if you believe this tree on the right, you believe that Glyciniales could potentially be recognized as an order that's phylogenetic, monophyletic. And then Hymenophylaceae is sister to that clade. Or, on the other hand, Hymenophylaceae and Glyciniaceae might be each other's sisters, which is what we found in the nuclear tree, in which case this order falls apart. Or at least you have to take out Glyciniaceae from the order Glyciniales. So important implications here for classification. So again, we found, I'm going to show you two first, and then I'll show you some additional ones. So there were two topologies amongst all these studies that found a monophyletic Glyciniales. So there we've got Hymenophyllales, sister to a group that includes other leptosporangiates and a clade that would correspond to the order Glyciniales. So here, the relationship is a little bit different, where we've got these all forming a clade together, but within that clade, you could recognize Glyciniales as sister to Hymenophyllales. So the key point here is that these two topologies are the two topologies that show support for this order as being monophyletic. And you can see there's a number of the plastid studies that agree with that. With that one, there's another more support from other plastid analyses for this relationship. These two, the mitochondrial analysis in pink and this one nuclear study, based on their taxon sampling, they would agree with either of these. They agree with this or another resolution. And here are the other resolutions that we find amongst all of these different studies. And so, again, those two would support either this relationship or this relationship down here. And you can see that the bulk of the nuclear support, well, all of the nuclear support essentially would go with a situation in which Glyciniales is not monophyletic. And the way those relationships fall out is a little bit different between these topologies. So there has been a proposal in the new version of PPG to not recognize the Glyciniales on the basis of these newer data, and it was rejected. So the Fern community really wants Glyciniales to be in order, and you could go either way. I think it's really a testament to the fact that there are no right answers in taxonomy or classification. There's what you consider to be the best answer based on the data that you have in front of you. So I can't remember whether I voted on that or not. I think I might have abstained from that vote. It's a community voting procedure using GitHub. So yeah, we got a lot of nuclear data down here that suggests, including GoFlag, that suggests that we should not be recognizing Glyciniales, but we will be hanging on to it a little bit longer in the official Fern classification system. So I've just got a couple more slides left because I wanted to say something about molecular dating. So we did do dating analyses on both the plastid and the nuclear trees, and I'm showing you those two superimposed on top of each other. So the blue tree is the nuclear tree, the yellow tree is the plastid tree. And you can see that there are a few places where the nodes along the backbone look a bit different, the ages and the dates look a bit different. And of course, those two topologies are different in several places, so obviously there are going to be some differences. But when I made this figure, and of course I was very careful to scale them correctly, and what really stood out to me was the similarity in crown ages between these two analyses. like some of these line up so closely that you can't even differentiate the lines from each other. So I thought that was quite interesting, that our ages for the crown groups of these families between the nuclear study and the plastid study aligned so closely with each other. And then the last thing I wanted to show you is another sort of way of visualizing the results from a number of these studies that I've been talking about today. So this is now switching to stem ages. So I was just emphasizing crown ages. This is switching to stem ages. And along the bottom here, you've got all of the families arranged more or less from oldest to most recent. And you can see, and so then these are the studies. And if it's a triangle, it's a plastid analysis. If it's a circle, it's a nuclear analysis. And then these are the two GoFlag analyses in green. And we did, for GoFlag, we used two different sets of calibrations. So this paper up here, Schiffpeltz and Pryor 2009, this was like the gold standard molecular dating analysis in Ferns, and they published a supplement to that paper that had just a beautiful breakdown of all of the fossils and how they assigned them and what ages they could be assigned to. And ever since, that's been the resource that people have gone to for secondary calibrations in molecular dating analyses of ferns. So all of these papers, except Nitta et al. 2022, used data from this paper as their calibration points. And so we did that with GoFlag, and that's these darker green symbols. But Nitta et al., they were the first to kind of go back to the drawing board. They evaluated some new fossils that had been published in the time between 2009 and they built a really beautiful, I think it's hosted on GitHub as well, actually, really beautiful data set of all of those fossils so that they added to the kind of the resources available for doing molecular dating in ferns. And so we also used their calibration set with the GoFlag data just to see how the two calibration sets compared. So if you can find the greens, I know it's a little complicated, but like here, they're right there, they're pretty close together, but in some cases they're a little bit further apart. And really what I want to draw your attention to is just like the enormous spread in some of these families in the ages that you find across all these different studies. So if you ever hear a fern person say with a lot of confidence, X family is Y million years old, don't believe them. Because, yeah, I mean, the minimum amount of spread, I think, is over here with Culcitaceae, and it's still like 70 or 80 million years of spread across all those different studies. So molecular dating is hard, and you can get very, very different results, basically, is my takeaway message from this slide. So with that, I want to thank all of the folks who were involved with GoFlag. So our PI group was really great. Funding from NSF was awesome. It obviously made this possible. And then I mentioned earlier that we used this sort of community-sourced approach to get all of the samples that we sequenced. And these are all of the people who contributed data for the fern analyses. So these people, none of this would have happened without their generous donation of data. And a lot of them have published papers on their own, families, genera, other kinds of questions with the GoFlag data. And with that, just thank you again for having me. I'm so happy to be here, and I'd be happy to answer any questions. Yeah, Rosie, yeah. I don't know if we got the bouncy thing to work, so I don't know, or I can give you my microphone.